In this video, we will be exploring the topic of gingival epithelium. Before diving into specifics, let's go over some terms that we'll come across in this lecture. What is stratified squamous epithelium? The name itself is giving us a clue. Stratified means many layers, and squamous is referring to flat cells. So stratified squamous epithelium is a tissue which is made up of layers of cells, and the topmost layers of the cell is flat. So in this type of the tissue, the only bottom layer of the cell is attached to the base, and this base is known as basal lamina. Other layers are sitting on top of each other. Gingival epithelium is a type of stratified squamous epithelium. Now let us see how do the cells grow and multiply in this epithelium. The bottom layer of the gingival epithelium is close to the basal lamina, this layer. So this layer is having some specialized cells which are known as stem cells. These stem cells are like a factory to the tissue, or they are also called as building blocks of the tissue. They are producing new cells by undergoing the division. So as the new cells are produced, they are pushing the older cells upward and closer to the surface. This is a basal lamina over which the basal cell, which is a stem cell, is present. So the stem cell is undergoing the division and it is producing the new cell. The new cell which is formed is pushed upwards. That means this is a stem cell and the newly formed cell is going and sitting on the top of the basal cell layer. And again, when the stem cell undergoes the division, a new cell is produced. And the older cell is moving towards the surface. So this is the stem cell. As it undergoes the division, the new cell is produced in between the older cell and the stem cell. So this is the new cell which is formed. So this older cell has moved upwards towards the surface. And again, when the stem cell is undergoing the division, the new cell is formed here and these two older cells are moved to the top. New cell is formed here and the two older cells are pushed upwards. When the cells finally reach the top layer, they are fully matured and they are ready to shed off from the surface of the tissue. Think of it like a cycle. New cells are formed at the bottom. They move up, change their shape as they go up, and eventually fall off once they reach the top. What is turnover time? Turnover time is the time it takes for a cell to be created at the bottom layer, move up through all the layers, and finally shed off from the surface. In gingival epithelium, the turnover time is 41 to 57 days. So it is taking about 1 to 2 months for a cell to complete its journey from the bottom layer to the surface. Next, let us see what are the types of mucosa. First one is masticatory mucosa. It is occupying 25% of total mucosa, and this type of mucosa is seen in the gingiva and the hard palate. And it is the part that is coming directly in contact with the food during the chewing. So this mucosa is keratinized in order to withstand the pressure. Second is lining mucosa. It is covering up to 60% of total mucosa, and this is the major type of mucosa in the mouth. So this lines the floor of the mouth, ventral surface of the tongue, alveolar mucosa, lips and the soft palate. Since it is not taking part in the chewing, this mucosa is non-keratinized. And last is specialized mucosa. It is covering 15% of the mucosa, and this is found on the top surface of the tongue, that is on the dorsal surface of the tongue. Stratified squamous epithelium can be categorized into two types: keratinized epithelium and non-keratinized epithelium. Now let's focus on the keratinized epithelium. Keratinized epithelium is organized into 4 distinct layers, each name based on its morphological characteristics. The first layer is the stratum basale. Above stratum basale is stratum spinosum. Above this layer is stratum granulosum, and the surface layer is called as stratum corneum. Each of these layers has a unique function in maintaining the barrier properties of gingiva. Let us continue our lecture by discussing about the basal cell layer. So basal cell layer is resting on the basal lamina. The cells in this layer are cuboidal in shape. And these basal cells have the capacity to synthesize the DNA, and they are also undergoing the process called as mitosis. So as they are undergoing the mitosis, they are able to produce the new epithelial cells. Hence, they are important for epithelial tissue renewal. Above the basal layer is the spinous layer. This layer is also having some mitotic figures. So the layer which is having the mitotic figures is known as parabasal layer, as it is present immediately above the basal layer. So now this parabasal spinous cell layer along with the basal cell layer together is known as stratum germinativum. The name itself is indicating that stratum germinativum layer is important for tissue regeneration. So let us study about the structure of the basal cell. What are the components of the basal cell? It has ribosomes and rough endoplasmic reticulum. These two organelles are important for protein synthesis. So the proteins which are produced by the basal cells, some of the proteins are responsible for the formation of the basal lamina. So this basal lamina is an important structure which connects the epithelium and the connective tissue. And this basal lamina is also an important structure in supporting the epithelium. Basal cells are also having the specialized structures which are known as hemidesmosomes. These hemidesmosomes are present at the bottom of the basal cell, and these hemidesmosomes are playing an important role in attaching the basal cell to the basal lamina. This is how the basal epithelial cell is attached to the basal lamina. And let us see how the two basal cells are connected with each other. So two adjacent basal cells are connected by a specialized structure which is known as desmosome. So the two basal cells are communicating with each other by a specialized junctions which are known as gap junctions. These basal cells are also having some structures which are known as intermediate filaments. These intermediate filaments are produced by the proteins of the basal epithelial cell. Let us have a closer look on the structure of hemidesmosomes. This is a basal epithelial cell. At the base of the basal epithelial cell, there is a specialized structure which is known as hemidesmosome. So this hemidesmosome is having the intracellular plaque. There are two components of intracellular plaque. The first component is this shaded portion, which is called as inner plaque. So this inner plaque is present towards the epithelial cell. And there is another portion that is present below the inner plaque. This shaded portion is known as outer plaque. This outer plaque is present towards the basal lamina. Now there are 4 proteins in hemidesmosomes. 2 are attachment proteins. And remaining 2 are transmembranous proteins. So this intracellular plaque is having attachment proteins. This plectin and BP230 are the attachment proteins. So these proteins are present in the intracellular plaque. So these two proteins are establishing the link between the intermediate filaments of the basal epithelial cell or the tonofilaments of the basal epithelial cell to the hemidesmosome. And other proteins are transmembranous proteins. BP180 and integrin alpha 6 beta 4 are known as transmembranous proteins. As the name suggests, these proteins are traversing from the basal epithelial cell. They cross the basement membrane of the epithelial cell and they reach the lamina lucida and stop there. Whereas BP180 is starting from the outer plaque of the hemidesmosome. It traverses the basal epithelial membrane of the epithelial cell and it enters into the lamina lucida and it is also entering into the lamina densa. So in the lamina densa, this BP180 is attached to another protein which is known as laminin. Whereas alpha 6 beta 4 integrin is also attached to the laminin at the junction of lamina densa and the lamina lucida. This is alpha 6 and beta 4. This portion is the alpha 6 subunit and this is the beta 4 subunit of the integrin. There are some anchoring fibrils which are attached to the laminin. So this is how the hemidesmosome is establishing the connection between the basal epithelial cell and the basal lamina. Let's now shift our focus to specialized structures called desmosomes. These are the vital cell adhesion structures which are serving to tightly bind the adjacent cells together. They are providing the mechanical strength and also stability to the tissue. These structures are especially important in epithelial tissues like gingiva, where the cells are constantly exposed to the mechanical forces such as chewing. The structure of the desmosome can be broken down into two main components, the transmembrane portion and the intracellular dense plaque portion. Let us discuss about the transmembrane portion first. This transmembrane part of the desmosome consists of two specialized proteins. The names of these two specialized proteins are desmoglein and desmocolin. Desmogleins and desmocolins, they are extending from the surface of one cell and interact with the counterparts of their adjacent cell, and they are establishing a strong bond between two cells. Adherent proteins are desmocolin and desmoglein. So these intracellular dense plaque is linking the cadherin proteins to the intermediate filaments or the cytokeratins of the cell. Let us discuss about the second portion, that is intracellular dense plaque. On the cytoplasm side of the desmosome, there is a dense plaque of the proteins which are anchoring the transmembrane proteins to the cell cytoskeleton. This dense plaque is having proteins like desmoplakin and placoglobin. Next is gap junctions. They play a crucial role in cell-to-cell communication, and these gap junctions are characterized by their low resistance. These gap junctions facilitate the direct passage of ions and smaller molecules between the adjacent cells. Through this form of communication, cells can coordinate activities like propagation of electric signals or sharing of metabolic substrates. This is allowing a synchronized behavior across the tissue. Unlike other epithelial cells, gingival epithelial cells are lacking the tight junctions. Now, the basal cells of the stratum basale are classified into two types, serrated and non-serrated. The serrated cells are heavily packed with the tonofilaments, which are the bundles of keratin intermediate filaments. So these filaments are essential in providing the structural support to the cell, particularly in terms of attachment. Serrated cells also have some specialized structures at the base, which are known as pedicles. These pedicles are the protoplasmic extensions of basal cells, which are projecting towards the underlying connective tissue. It plays an important role in the attachment process. The non-serrated cells, on the other hand, do not have these specialized structures called pedicles, and they are less involved in the attachment process. They are more focused on the regenerative and protein synthesizing functions. Non-serrated basal cells give rise to two distinct groups. One is slow dividing cells and second group is amplifying cells. Slowly dividing cells are important in protecting the genetic information of the tissue. By dividing at a slower rate, they are reducing the chances of errors in the DNA replication. On the other hand, amplifying cells have a different role. These cells are involved actively in the process of increasing in the cell numbers. They are undergoing the faster rate of cell division when compared to that of the slowly dividing cells. so which enable them to contribute to the maturation and also the renewal of the epithelium. By this, we conclude our discussion on stratum basale. If you liked the video, hit the bell icon below and subscribe to my channel. In our upcoming videos, we will dive deeper into other layers of gingival epithelium, exploring their unique characteristics and roles in maintaining oral health. Thank you for watching.